Welcome to Dr. Carr Society Mission. So this is short lectures or tutorial on RBCs and NWBC, that is the cells in the blood, how you can estimate their number and all that. So this is very simple mathematics that we're going to take you through. Right, so let's assume here this is the vein. Okay, we take our blood sample, we take 20 mils using the syringe. Now we've taken 20 mils. Now we empty it into a beaker, or let's say into a test tube. Okay, test tube is usually what we use. We empty this into a test tube. So this is 20 mils. Now we want to determine the number of red blood cells or WB white blood cells in this sample that we've taken. Let's see how we can go about it. So the next step that we'll do is, this sample is too much. It's 20. We can't, count all the cells in 20. So what we'll do is we'll take sample. That we take part of this total uh, blood that we've collected. So let's say we are taking half of that. I'm just assuming for simplicity or to make the calculation simple and then make it intuitively easier to understand. So let's say we took half, or we take half of that. This is 20. If we take half, that is 10 mils. Okay, now we take 10 mils. Now with this 10 mils, what we can do further is we can dilute this 10 mils. We dilute it further. Okay, we can dilute this 10 mils. We dilute for several reasons. Okay, now if let's say we have 10 mils and then we add 90 mils to it, that will be the total volume will then be 100 mils. So in essence, we have diluted this 10 mils to 100 mils. That is, we've diluted it. We've increased the volume by 10 times. Okay, so the dilution factor is 10 here. Okay, note it very well. So the final volume was 100. The initial one was 10. So the dilution factor is the final divided by the initial, which is 10 factor. So it means if whatever number of cells that we have there has been diluted 10 times. Okay, so the concentration has been diluted 10 times. That is one thing we should note. So that's the dilution factor. Now let's get back to business, where we want to know the number of cells in there. So here you see that I'm just creating a replica of what is above here, so that we have it. So let's say the 20 mils that we took had 6 RBCs, or red blood cells, had 6 of that. Now that is it, just representing it here. If we take half of this, we'll have 10 mils, and then we'll expect that all things being equal, because this is containing 6, if we take half, we should have 6. We should have 3, half of the 6 in this sample. So that's what we have. Now, if this 3 is in 10 mils, now if we dilute this into that's 10 mils, we dilute it to 100 mils, we will still have the same number of 3 cells in this huge volume. Do you get it? Now, what I'm doing, or what I have done here is very important. It's just trying to illustrate this pictorially so that you can appreciate the maths. So here we have 3, which is the 10 mils. This is the 10 mils, and these are 90 mils without any cells. Okay, so when we add this together instead, we're going to have the 3 distributed within this 100 mils. Okay, this is it, right? So let's, for simplicity sake, I will just say, okay, let's take 1/3 of this volume, which is about 33.3, to be 33.333. So let's say 33.3. If we take 1/3 of this volume, we would expect that we'll have only 1 cell in it. Do you get it? And that is what we have. So let's see, that's what we have. If we take 33 mils, we will have what? 1 cell in it. Okay, so 33 mils will have 1 cell. Therefore, 100 mils will have how many? It's just the dilution factor, and the factor that you have times the cell will give you the total cell number, and that is giving us 3. Do you see that? Right. So where do we have that? Let me see. Sorry for bringing that. Sorry, that's the concentration. If we are looking at the concentration, not the number, let me leave it here. So there is the dilution factor we are looking at. This is the dilution factor. But what we can say is if 33 is equal to 1, that is what we've done here. We'll come to that side. But there is a dilution factor that you can get. The dilution factor is 10 here. Now let's assume here. Let's look at something. But I mean, if you are not looking at the dilution factor, you want to do simple math. If 33 is 1, then 100 will be, then you know it will be 3. That is also 1 there. Now let's working things backwards. Okay. Now that is getting these things backwards. Now, so let's say we have a sample. You are told, instead of putting this, you took a sample, then you take a small amount of that sample, you dilute it. You may have this, what you call the grade. Okay, then maybe you pour a certain volume of this on the grade. Maybe that is 1/5 by 1/5. That is the volume. Okay. So you know volume is equal to length times breadth times height. Okay, so we can calculate this volume. Okay, so long as we can get this volume, if we have this volume, we can estimate uh, how many cells that will contain. Okay, because we know the amount that was in this. Okay, right. Now even if we did not know, we know at a point, this is where we are going to probably use our microscope. Okay, to see. So we are now going to use the microscope. Then we count the number of cells in here. So let's say when we counted, we had 1 cell in here. Okay, from this diluted one. Okay, so after diluting 10 times, we have 1 cell here. So then the total volume in this one, if you want to know the total volume in this one, you multiply by 10. It will give you depending on the volume here to the volume here, the ratio. Okay, the ratio between this volume and this volume. Whatever we got here, if we multiply by that ratio, it will give us the total number of cells in this. So this is more of simple mathematics. Okay, so that proportionality is what is being used. So let's say if we have, let's assume for simplicity's sake, let's say this is 2 mils. Okay, and then maybe we found 1 cell. Okay, right. Now if this is 100 mils and 2 mils is giving us 1 cell, then we can estimate how many cells will be in 100. And then when, let's say, so 2 mils is proportional to 1 cell. So 100 mils will be what? More. If more, let's divide. So 100 divided by 2 times 1 cell, and that is going to give us 50 cells. Okay, so it means if we have 1 cell in these 2 mils, then this total one had 50 cells. Now these 50 cells that we have here, remember the 50 cells that we had here are all coming from this 10 mils that we took from the sample. Okay, so you should have the picture in mind. If you have the picture in mind, you'll be able to understand this. Okay, so it means the 50 that we are talking about is here. All these 50 are coming from this small one. So it's the same as saying these 50 cells are in the volume that we have here, 10 mils of the first sample. Okay, once we know 50 cells are in 10 mils of the first sample, we can also work back to tell how much was in the total sample that was taken. So if 50 is in 10 mils, then how many cells will be in 20 mils? So times 2, then that will be 100. So we can do that even if we, after getting the sample that was taken here, we can even estimate how many cells will be in the body here. Okay, right. So we can even, if we know the total volume of blood in humans, okay, was about 5 liters. Okay, then we can also use that to get the total number of cells in that blood volume. And if you want to have the concentration, then we will know concentration will be equal to volume, number over volume. So once we know the total number of cells, we know the total volume of human, then we can get the concentration. You get it? But remember the dilution rule. Concentration 1 times volume 1 is equal to concentration 2 times volume 2. This is very important equation you may need. It's a very important equation you may need. All right, so I think this is the summary. Let me just go over. So you take sample 20 mils, you pour into a beaker or pour into a test tube. Now you take sample, you take a small bit amount of that amount here into this small one. Now this the one that you are going to dilute. You dilute into this, okay, and then take maybe another small sample here. Okay, so this final sample that you have here, this is the one that you are going to look, examine under the microscope. Okay, so what you examine under the microscope, you count the number and then the moles that it is found in. Once you know the number of cells you are getting, the moles that it is found in, you can estimate how much was in the total diluted one. Now remember, all the cells that you are going to get here are coming from here. So once you know the total number, it means this total number is the same number in this volume. So you can even talk about the concentration as well as the number in here. Once we know the number and the concentration here, we can estimate how much will be in this one, because it's whatever is here by a proportionate amount, we can get the total number of cells. If 50 are in 10 moles, then we can tell how much will be in 20 moles. We can get that. Once we get that, we are done. We can even extrapolate again. Go back again to get the number in the body if we know the volume of blood in the body, and also estimate concentration. Right. Thank you very much.